Okay, now let us look at lipid. Okay, what are the types of lipid tests? In general, there are two different types of lipid tests. The first test involves qualitative analysis, and the second type of lipid test involves quantitative analysis. Example of qualitative analysis in testing the presence of lipid are solubility tests, emulsion formation, Lieberman Brochard test, and Sudan stain test. Okay, now we look at quantitative analysis. There are several different types of quantitative analysis to test the presence of lipid. The first is we call it as the chromatography analysis. Second, saponification value. The third is the iodine value or iodine test. And lastly is the direct mass calculation. If you have watched the video which I uploaded in YouTube, it is regarding the method of quantitative analysis to test the presence of lipid using direct mass calculation. What's the difference between qualitative analysis and quantitative analysis of lipid? Qualitative analysis of lipid could only determine the presence or absence of the lipid, while quantitative analysis could actually test the presence of lipid in any samples, and it could actually determine the value. The value could be either in number or percentage. Example of qualitative tests to test the presence of lipid in any samples could be conducted using the brown paper test. As what you can see from the picture, if oil is present, it is indicated by a spot which is translucent. This is a very simple test that you can perform in the comfort of your own house. Okay, now we shall look at the aspect of direct mass calculation. The direct mass calculation method to determine lipid present in food sample could actually quantify the value in either a number or in percentage. This is the experiment. This is an experiment that is called the direct mass calculation, which is a quantitative method to measure the presence of lipid. Not only that, it could actually give you the value either in the form of number or in percentage. This is a relatively simple experiment where you need to measure the weight of the apparatus that is used, which is the beaker and the glass rod, together with the dried food sample. And according to the protocol that is listed in your laboratory 5 on lipid analysis, the next step will be adding petroleum ether into the flask. The next step will be decant the petroleum ether from the beaker, and we try to dry out the sample but thoroughly by heating up on a hot plate. And finally, what you need to do is that you will weigh the weight of beaker together with the glass rod and the weight of the samples that is dried. And you can determine the percentage of lipid being extracted by using the formula that is shown under table 2, which is weight of lipid extracted over weight of crushed food sample times 100%. This is an example of quantitative method to test the presence of lipid in food sample, as well as you can quantify the lipid by a value. The value could be either in number or in percentage.